So we've covered the cell membrane, and we said these things are lipids, phospholipids, and we know that these phospholipids they make up this membrane that gives it these two different qualities. And these qualities are very important because they determine the function of the membrane, and then that determines the function of almost every cell organelle, because a lot of cell organelles have membranes. And we'll talk about the organelles today or tomorrow. Then we know that, so what are those qualities? You should know them already. I'm going to model them now. I'm going to explain them now, but everybody should have index cards with them. Can someone close the door? And I'm just going to make sure I point them out. Oops, that's too wide a line. This is hydrophilic. Because there's water outside and inside the cell, and that's true because this is a double, right? A bilipid layer. Bilipid. 2 layers. So it's a membrane is made of 2 layers of phospholipids, correct? Everybody okay with that? So to be clear what I mean, so no one's confused, we have 1 layer of phospholipids, 1 layer of phospholipids here, and another layer of phospholipids here. So 2 layers, almost like a sandwich. Now, hydrophilic, and then the other quality that the membrane has is hydrophobic. And you can think of the membrane as having the center of hydrophobicness, of hating water, and this outer area that is hydrophilic, that loves water. And that explains why the heads always point towards the water and the tails always point towards each other. But that also goes to this idea that we've discussed already of like dissolves like. And we discussed that, look, we need ions. We looked at this at a neuron, I believe, and we know we have these sodium ions and these chloride ions and these potassium ions and these bigger molecules called sugars that are polar. So this was big and polar. And these are small, but they're charged. The only thing that's going to get through this membrane easily, are little molecules like like oxygen, that can go right between the molecules of phospholipids because they're nonpolar and small. That seems like it's too much writing there, so I'm just going to put nonpolar and small down here. And of course, other molecules like CO2 can go right through the membrane because they're nonpolar and small. Now I'm going to go ahead and just put a little asterisk by those. Now, this is big and polar. This sugar molecule is big and polar. Also needs something else to help it through because it can't get through the membrane either because it's big. The problem it has is it's big and it's polar. If it's big and polar, it's going to be hard for it to get through the membrane. And if it's small and charged, it's going to be hard to get through the membrane. Small polar, like water, can get through the membrane, but it happens very slowly, and it can go both ways. Everything can go both ways, by the way. It's not just a one-way movement. So that's for non-polar and small, for polar and small. That's what we call permeable. You don't need any help with these molecules. But anything that's polar and big or charged you're going to need help. So what are some examples of polar and big, or just big? Any protein is going to be big. Any sugar is going to be big. Any sugar. Polar and any nucleic acid. It's going to be too big to fit through the membrane. Charged examples, we already have some: sodium, chloride, potassium, anything with this full negative or full positive charge. Anything that has that going on is not going to get through the membrane because it's charged. Okay? So how do we get those through? We need a protein. We need a transmembrane protein. It's called a transmembrane protein. This is your window or your door. And a transmembrane protein can look something like this. So this is a channel protein. It's going to allow specific things in and out, and it acts as a door, right? Transmembrane proteins act as doors or windows, right, into the cell. They can also act as signal mechanisms. So you can signal things using transmembrane proteins. So transmembrane proteins are not limited to being doors or windows, but that is a big part of what they do. And that's the stuff we've covered so far, and we should know this by now, right? This is not new. This is stuff that we should all have in our notes. We should have index cards. We should be making sure we got. Just trying to clarify, because some people were saying that they were a little confused over the notes, right? And their notes looked a little confused when they went back over it. So now this is pretty straightforward, yes? What's confusing about this? Go ahead and let me know. Look at it, and say, okay, I'm confused about this. Anybody? Look at it and tell me. Now's the time to speak up before the test. You're going to have a quiz next week. This week, you have a quiz this week. Next week, you have a small practice test. Come on. Look at it. Tell me where you're confused. Zoom in on permeable? Sure. It says the stuff that's permeable. Remember, what does permeable mean? Who remembers what that word means? Anybody? Anybody? You really have to watch Ferris Bueller's Day Off because I feel like that's one of those teachers in this particular morning class especially.
It allows things through.
Thank you. Allows things through is correct. And of course we're talking about molecules, right? Permeable means it gets through. This stuff over here is not permeable. This stuff needs help. And needs help. Without help, it can't get through. That's what it needs, this protein right here. It needs number one. If you're not gonna, it's not gonna, and remember, this is a single protein. I want to remind you, it's hard to remember when you're looking at it, but this is a cutaway. So if you were looking at it from top down, all you would see is a bunch of phospholipid heads. And then from the top, you would see the protein would look something like, something like that. And then the hole would be in the middle, allowing things to go through. Does that make sense to you? Sure, I can zoom into anything you like. I'm also recording this so we can go back. So if you see this hole here and these blue heads here, if you were looking down on it, it would look like this, a bunch of blue heads and then the holes in the center. See, that's how it acts as a door to this, to this cell that's round, right? It's a round cell. With all these phospholipid heads on the surface. And these proteins are floating around as doors to letting the inside and outside communicate, letting the inside and outside transmit molecules, let molecules out, allow molecules to come in. Because if this cell's alive, it needs to eat. So it needs to bring in sugar, it needs sodium, it needs potassium, it needs chloride, it needs water. So you need these holes to allow them in and out. How do you create a hole and still create the division? You have proteins, transmembrane. They're called transmembrane proteins because they allow, trans means to cross, right? Trans means to cross. So this means what? To cross the membrane. Proteins that cross the membrane. Does that make sense? No? Okay, gotta help me out here. What's confusing? Yeah? Yes. What here is confusing? Anything. Just point one thing out. Anyone. Everything is not an answer. Okay, I hear you, but I think that when you say everything, I fear that you're just being general. You're just generalizing. When you look at that, do you see where there is a protein? Do you know what a protein is? The word protein. Have you heard it before? Yes. Okay. You know it's a bunch of amino acids in order that form a structure, right? You remember amino acids? It's a polymer of amino acids. Amino acids is a monomer, it's just a bunch of amino acids. Yes? And you know what phospholipids are? Right? Yes or no? Phospholipids. The blue with the yellow. That's right. Like we said, you need to go over that. I've been saying it for a week now. You need to memorize these things. If you don't memorize the very basics, it's like trying to read without knowing the letters. Of course you can't read.
Isn't the protein the one that you have labeled 1?
Yes, that would be the protein, the one that's labeled 1. That's the example of a protein. It's just a sketch. Proteins are these long chains of amino acids. Some can be polar, some of the R groups can be polar, some of the R groups can be nonpolar, so that allows the protein to stick out and stick in. They're amazing little structures. So if you don't remember or know what a protein is, you got to go back and memorize that. Phospholipids, you got to know what those are. Or this membrane's not going to, you're not going to understand this at all. Then I agree with you. You understand nothing of what I said if you don't understand protein and phospholipid. So you understood it when we went over it the first time, but you didn't memorize it. So make up index cards, get tutoring, but you need to get that in your head, because if you don't understand those two concepts, I'm afraid it's going to be very difficult to answer the problems on the exam. So, yes? It's Unit 2, Unit 3. Look at the syllabus I gave you. I gave you a syllabus. I explained everything. I sent it to you. I need to print it. I haven't been able to print it yet. Who knows where the photocopiers are in the office? No? Okay. No, don't ask. Do you know where they are? There's two of them. You know where both of them are? Yeah, and then the one that's in the room. In the teacher working with the desk. All right, go in the one with the back room. There should be a stack. In the teacher room. There should be a stack of photocopies of your work. You asked me what the problems are going to look like. I already printed sample problems for you. So they're not going to be easy if you don't understand these concepts. All right. Now I have to go on, so if you're confused, I need you to write down questions. Identify what you're missing. I already identified two problems we have here. Problem 1, you didn't know what a protein was. Problem 2, a phospholipid. So those are things you need to write down, things you don't understand. Things you don't understand are even more important than things you do understand. Yes, Miss, yeah. Sure. And I Bolden to the office, please. Is she here? Where is she? There you are. Could you take the pass and come back? I don't know why she needs you, but she needs you. So you need to know this. Okay? Then identify what you don't know, because that's even more important than what you do know. Often it's what you don't know that gets you the wrong answer. So that's the membrane. Today you're going to have an activity that is going to have you look at a cell and tell, is this stuff going in? Or is it going out? You're going to be looking at things called two processes, one known as diffusion, another known as osmosis. They're pretty much the same thing, but we'll talk about that in a minute. Now, diffusion is exactly what you all were some of you were saying. You were smelling something in the hallway. That's what somebody was saying earlier this morning. Something sour. Now, you didn't know where it came from. You weren't sure what it is. But do you know a dog could smell miles away? Because from someone, if you were miles away from them, they can smell your scent and follow it. So let me give you an example. So if you have a dog and a little boy's lost, and the boy's here. I'm just going to use a dot. I'm not going to go through trying to draw a little stick figure because everybody's going to make fun of me. But over here's your dog, and the kid's lost in the woods. And here you are with the dog. And the dog's trained, and you give the dog a sock, the kid's sock. The dog is going to be able to use his nose to sense molecules in the air. You call that smell. And around the boy, there's a lot of these molecules because it's close to him. They're highly concentrated. What's going to happen is I move away from the boy. Either you get more concentrated or stronger. Does smell get stronger as you get closer to something or weaker? Stronger. So these are weaker. They're further apart as you get away from the boy, right? Do you agree? And the boy could be miles away. But these molecules that he produces, you do this too. You produce molecules. You have a smell. You have a very specific smell. Very specific smell. Very specific combination of molecules. You and I, you probably know the smell of your mother. I know I do. Sometimes I think of it when I'm alone and I just think about my mom. I know my wife's smell. You probably know the smell of a few people, correct? Because you get close to them, you love them, you hug them, you kiss them. You know what they smell like, right? Yes or no? Yeah. And I'm not talking about cologne. I mean, if you use the same cologne as someone else, right, they can smell similarly. But that too, cologne is part of it as well. It's a smell that we put on artificially. So the dog, though, can smell much more detail. Their noses are much better at smelling. And what you'll do, you'll see a hound dog do this. They'll come around, they'll go back and forth. What they're doing is looking to see where's it more concentrated. Oh, there's more of these. It's stronger over here, this way, this way, this way, this way. And all along, as soon as he picks up the scent, yeah, not yet. It'll howl. It'll say, oh, not yet, not yet, not yet, not yet. Don't go yet. Don't go yet. Is she still here? I swear I heard her keep going. So anyways, it'll howl and then keep going. Thank you. And keep going towards the scent. And they'll find the boy. That's how they find boys. That's how dogs help find you. Dogs can actually sense when you're about to have a seizure. People that have epilepsy, when they're about to have a seizure, they let loose stress hormones into the air. You and I can't perceive it. No human can. But dogs can sense it. They have something called microtremors. Dogs can sense the microtremors, and they can warn someone that a seizure is about to happen. And we have these dogs that you'll see walking around with people that have special vests, and they ask you not to pet them. Have you ever seen those dogs? Yeah, those dogs are trained to help people that have seizures, help them know when a seizure's coming, and they can prepare for it. But they'll sit next to the person who's having a seizure. They'll even have a little sign that'll say, please don't disturb, let the seizure pass, or whatever. So these dogs can be very helpful. So the point here is that the way the dog senses smell is this thing. This thing here. This is another of these vocabulary words, guys. If you don't get the vocabulary words in your head, and I know there's a lot, you're not going to be able to answer questions. But this thing here, where you see you go from high, do you agree that's high? To low over here, where you go from high to low, it's called a concentration gradient. So, anybody here do any drawing? Okay, excellent. So, when you draw, you have this technique you use where you do this. You start to draw a lot, very dark, right? And you start to pull away from the paper, and it's lighter and lighter and lighter. And lighter. Anybody who's an artist does this. I know I took a few art classes. I suck, but I keep trying. This is called a gradient. it's going from darkest to lightest, right? Does that make sense? So that's what the word gradient means. The word gradient means to go from dark to light. Or not dark, from a lot to less. Whatever that means. Is that making sense to everybody? All right, so concentration gradient is where you have a lot, high, and then on the other end you have low. And because you have that direction, a dog can find you if you're lost. Okay? Also, you'll see that what people will do if they're running away from dogs, they'll go in the water because the water helps hide your smell. It doesn't help that much though unless you can stay underwater for a long time, because as soon as you come out, your smell's going to start again. As soon as you breathe, there goes some of your molecules. Yes, go ahead. I just wanted you to get that, what I just said. Are we all good? All right. So that's a concentration gradient. Now, everything in the universe, electricity, light, gases, heat, everything in the universe, hear me clearly, moves from high to low without any extra energy. Period. Everything moves from high concentration to low concentration. This explains the wind. Where you have high pressure, wind will move from high pressure to low pressure. When they put high and low in the maps during the weather on the news, they'll put H and L. H means high, L means low. That's the direction of the wind. The main direction. Obviously, wind can go in different directions depending on what it hits while it's traveling. But high to low, everything moves from high to low. Period. When you have a barrier, if the barrier has spaces, obviously if this has no spaces, nothing can get through. But if it has spaces, here the ions are charged, okay, and here this blue, it's in water, so these are solutes in water. And I'm going to go ahead and just do this. It's all in water, okay? And if I have these ions in water, these are, let's say sodium, and there's a sodium ion on the left, and then you have also maybe some sugar molecules. Use a darker color. Why am I drawing it as a hexagon? Because glucose is what? Is a hexagon. We should all know that by now. This should not be a surprise. 6-carbon sugar, we know that. We know that's going to be on the test, so that's not going to be a problem for us. We know we have to know our carbohydrates, our lipids, our proteins. Mendoza told me that I needed to know that, so that's not going to be an issue for me. So you have this combination of sugar and sodium. Where is sodium the highest? I'm asking where sodium is the highest. Sodium is the plus charge. And sugar. Is the sugar going to pass through? If this is a phospholipid membrane, is the sugar going to pass through without help? Will sugar get through the membrane? See, the yes, no is a bother. Let's go back to what we said originally. It should be in your notes. You should have written it down. We said what? Big polar molecules need help. They cannot cross the membrane without help. That includes proteins, sugars, and nucleic acids, among others. Correct? They cannot pass through. Also, ions can't pass through. You have to know this or you're not going to be able to answer the questions on the OST or here in class. You will not, and you can't tell me you forgot it because it won't matter if you forgot it or not. If you don't remember it, you won't. So sugar normally does not pass through. But this is not a phospholipid membrane. This is just a synthetic membrane. Sugar is not going to pass through. It won't let sugar through. This is we call it a dialysis bag. We have some of it here. We're going to use it in the lab. You have a dialysis bag. So it's a bag that's used in dialysis. You know what dialysis is? Is when you move the blood through a bag and then the water gets through, but some other stuff doesn't, and you're able to filter out. So the bag has these little holes, these tiny little holes. It won't let sugar through, but will let ions through, small ions. So this will get through. Is that clear? So the first step to these kinds of problems, these diffusion osmosis problems, the first thing you got to do is write down what is or is not permeable. What is this membrane? What does or does not pass through this membrane? In this case, this is the first question you're always going to ask. Should I simplify the question? Let me simplify the question because I don't like the way that question sounded. Let me make it easy. What does or does not pass through this membrane? Right? That's the question. What does or doesn't pass through the membrane? It's as simple as I can put it. So the answer here, there's two solutes. I would have to say sodium does what? Pass through. And glucose does not. You have to write that down because you will get confused on problems, especially in the early parts of these problems, but specifically, even if it seems simple to you, write it down. 2, the next question you have to ask yourself is where is it? The stuff that can pass. Do you care about the stuff that can't pass? Not really, because it can't pass. It's not going to go, whatever it is here, if this side's high, this side's low, it doesn't matter because it's not going to be able to get through the membrane, right? The question you're trying to answer is in what direction are these substances going to move? Right? I should have written that question down first, but sometimes I jump ahead of myself. I apologize. So the question you're trying to answer is, what is moving and in which direction? Is that clear? Now, to be clear on why this is moving, I know some of you are not going to care, you're not going to remember, but I'm going to say it anyways. Kinetic energy, everything is moving all the time, it's vibrating. Now, as these vibrate and they go this way and that way, and they're moving in the liquid in this little chamber over here on this left side, as they're moving around, the sugar hits this wall, it can't get through, it bounces off and moves in that direction. So the sugar is just going to keep moving around in this space. This sugar is just going to keep moving around in this space. Does that make sense? But this ion, they can keep bouncing around. Eventually, one of them is going to do what? Will go through. And randomly, this ion could easily go through in this direction. So they can move in either direction. But if I have a lot of them on this side, if I have it high over here, what are the odds? What's going to happen over time? Can somebody tell me? Eventually, what's going to happen? That's right. These positives are going to start moving in this direction. So they're going to move. This one might randomly move over here. So you got 2 to whatever it is, and then 3. And yeah, some of these will move over here, but it's because there's more on this side, you're going to have more movement towards the low. You get high concentration moving to low. Is that clear? So high to low. So the second question is, where is it moving? Well, I'm not even going to bother answering the sugar one because the sugar does not move. But the salt, is going to move from left to right. Correct? Now, why is the salt moving and not the sugar? The sugar can't fit. The salt can. This is not a membrane.
You said the sugar can fit.
The sugar cannot go through. We said that, didn't we? We all agree that we said that a minute ago? Okay. So we've identified that the things are going to move from high concentration to low concentration. And I was going to give you this, but I don't think you're ready for it, so I'm going to wait. I'm going to wait to give you these. All right. Do we get that so far? All right, I'll be posting these. Please make sure you write down these ideas and get them in your head, practice them, quiz each other. You have a quiz this week and a small test next week, and then you have your final coming up.